Happy Friday. That covers you whatever time zone you're in. I don't think anybody's yet into Saturday or still lagging behind in Thursday. So as chair of this study and of this committee meeting, I'd like to welcome everyone. The task being undertaken by this committee of the National Academies of Science, Engineering, and Medicine is to examine the long-term medical and economic impacts of increasing antimicrobial resistance in the United States. The task being undertaken by this committee includes commenting on the progress against the national strategy of combating antimicrobial resistance, and the full task is described on the study website. I want to note this is an open on-the-record session. Interested individuals and members of the press have been invited to attend as observers. This is not a news conference, however, and we will not be entertaining questions from the floor. Reporters who would like to speak with committee members are kindly asked to touch base with the study director, Jillian Buckley, and her contact information is on the study website. This is an information gathering session. The committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, conclusions, and recommendations. Therefore, I ask that everyone here today be extremely mindful of the fact that the committee has made no conclusions, and it would be a mistake for anyone to leave here today thinking otherwise. Comments made by individuals, including committee members, should not be interpreted as positions of the committee or of the National Academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views nor those of the committee. The committee will deliberate thoroughly prior to writing its draft report. Moreover, once the draft report is written, it goes through a rigorous review by experts who are anonymous to the committee, and the committee must then respond to that review with appropriate revisions that adequately satisfy the National Academies Report Review Committee and the National Academy of Sciences president before it is considered an official National Academies report. I would, at this point, like to welcome our speaker this morning, Dr. Dantas, who's going to speak to us about an issue we were discussing earlier this week about using predictive genomic information. So Dr. Dantas, I'm going to turn it over to you. Hopefully you can, there we go. That looks good.
Sorry, I need to turn my mic on first. All right, there we go. So yeah, thank you for giving me this opportunity to chat with you this morning. Thank you all for what you're doing in this committee, something that's dear to my heart. And so before I get started, I want to make sure I'm here as a representative of an incredibly talented and diverse group of individuals. And I'm here merely as their spokesperson in terms of presenting their research and their ideas. And before I get into the science, I just want to make this very, very clear that we welcome and champion diversity of opinion and people. And in this particular time, I think it's really important for us to note that infectious agents in particular have this dangerous possibility of being misinterpreted. And so I would just like to say, because I'm going to be talking about bacteria, because I'm going to be talking about antibiotic resistance genes, that one recommendation, and this is a personal recommendation I would make, is that we stop naming infectious agents after people and places because there are dangerous consequences. So with that, I'm going to switch to telling you a little bit about what we do. For some context, our lab is deeply interested in both understanding and predicting how microbial communities function and act, how that places us as humans in the context of the broader world, how that translates into understanding and diagnosing infections, distinguishing between the so-called good and bad bugs. We also have some work in trying to adopt the language of microbes as opposed to the language of warfare in terms of using microbes as part of our strategy to combat infectious agents. And then what I'll be talking to you about today is how do we potentially harness new tools, both wet lab and dry lab, so that is both bench experimental as well as computational, to perhaps ask old questions in new ways. So This is one cartoon way in which our lab views the world in terms of these artificial names that we as humans like to place on our various habitats, and names which microbes certainly don't care about. And so really this talk is partly about recognizing this idea that despite the fact that we would like to compartmentalize where we live and where our waste is produced and processed and where the environment is, microbes can trivially traverse across those habitat barriers. And selection pressure, such as antibiotic use in one particular habitat, can easily select for antibiotic resistance and other features in these microbes, which can then transmit across those habitats. And really today, I'm going to give you a couple examples of what that means, looking at it at different resolutions of asking how these selection pressures play from environment to clinic and back. So this is something that you've probably already talked about in your discussions, but just as a review, we obviously realize that one way in which antibiotic resistance arises is through this process of vertical transmission, the Darwinian process where as cells divide, mistakes are made, and then every once in a while, a mistake can be beneficial. So as shown here, maybe a mutation in the genome of this organism allowed it to evade an antibiotic impact, and that can sweep through the population. However, from genomic studies and mechanistic studies to follow of infectious bacteria in particular, horizontal gene transfer, this parallel mechanism of resistance, appears to be a dominant mode to allow the dissemination of antibiotic resistance, right? So various methods that bacteria have to borrow and donate and pick up antibiotic resistance genes from each other. And this appears to be a pretty quick way to then take something that maybe some other bug has already evolved and transmit over. And a lot of the methods that I'll talk to you about today are a bit focused on the idea of how do we capture these horizontal transmission events and risks, perhaps before they've even occurred. So how do we perhaps harness bellwether events in environmental microbes that tell us what might happen in the clinic? So let's talk about methods available to us for studying resistance. I'll use the word antibiotic resistome, coined by Jerry Wright about a decade or so ago, maybe a little bit more, to refer to all genetic elements in the study of interest, could be a group of microbes, cultured or uncultured, that give them the ability to resist antibiotics. So one way to harness the resistome is old school traditional microbiology. You domesticate the animalcules, as Leeuwenhoek called them, and then phenotype them. And certainly in the incredible era of high throughput everything, this is one certain way in which to very specifically associate a genotype with a phenotype. And now in this day and age of being able to sequence anything under the sun, now on USB chips over the order of minutes to hours, you can certainly predict, or you could propose that you could get everything else, the bugs that are not cultured, by just sequencing them. And this certainly gives you a nice complementary almost engine for finding antibiotic resistance genes initially in cultured bugs, and then using sequencing them to infer what happens in the other bugs. The problem is this particular engine can only iterate on the known. So that is to say, if you were to sequence a sample and find DNA elements that you want to call an antibiotic resistance gene, in this scheme, the only way you'd really be able to do that is to look at the genes that were previously identified using culture-based methods. And many people, and our lab is part of that whole, have shown that that is a vast underestimation of this so-called antibiotic resistome in virtually any habitat. And so what I'll introduce now is a method called functional metagenomics, or functional metagenomic selections, a method that was described first in the late 90s by Joe Handelsman and John Clardy and colleagues as a way in which to perhaps expand this part of the cryptic antibiotic resistome, the part of the resistome pie that perhaps is not accessible trivially through culture. And the hope I'd like to propose is that all of these are complementary methods, and eventually, perhaps we'd like to put even this functional metagenomic selection scheme out of business by having robust enough computational methods that can make de novo predictions. So until then, what is functional metagenomics? So at the simplest level, it's a high throughput cloning and selection assay. So you start out with DNA from a group of organisms. It's called that the metagenome. This is the collective genome of all of those organisms. You go through a cloning process. This is like any kind of DNA cloning, except rather than identifying or a priori defining what the genes are going in, you randomly break this DNA up. you put it into some sort of expression system, which will drive the expression of whatever's on that piece of DNA. And then you clone it into an easy to use host. We happen to use E. coli. You can use other organisms. We use E. coli for two reasons. One is because it is an ideal model organism with this Jekyll and Hyde feature. It can live as a commensal, a good bug, if you will, in the guts of humans, but it can also cause disease depending on the strain. It's also the most genetically tractable organism on the planet right now. So once you've made these metagenomic libraries in E. coli, each individual E. coli cell has a random chunk of DNA. And then you take all of them at the same time and go back to traditional microbiology. Plate those cells on agar plates that have antibiotics in them. The only thing that survives now are those E. coli that have a new antibiotic resistance phenotype, because you're plating them on concentrations where the original E. coli didn't have resistance. So now you have en masse identified a large selection of genes from this original metagenome that are able to, through a mock version of horizontal gene transfer, give E. coli the ability to be resistant to those antibiotics. And then, of course, the devil is in the details of going through and identifying what those genes are. It's that devil in the details that we've been spending the last about 10 or 12 years trying to figure out by marrying that basic engine, which I've shown again up here of the cloning and selection, with next generation sequencing and computational methods. So the main power of next generation sequencing is scale. It's the ability to do large numbers of sequencing experiments basically at the same time in the same single pot. So here shown is, an n number of selections, which you could do from different sources of microbial DNA, so different fecal samples of different groups of microbes, against a variety of different of your favorite antibiotics. You can use any antibiotic you want that would kill the wild type host. And then once you've got those selected fragments out, you turn to your favorite next generation sequencer. I'm showing this using Illumina short read sequencing, where all of these particular fragments have been cleverly first barcoded. Little individual unique pieces of DNA have been added to the fragments that came through this, such that when they go onto the sequencer and come out, the first thing that's read is one of these barcodes, which tells you, aha, barcode blue in this particular case came from selection 1, which happened to be a 3-year-old child's fecal sample that was subjected to penicillin selection. And then through this huge sort of demultiplexing experiment where each individual set of sequence reads are treated individually, and then you go to various computational methods that stitch everything back up and give you a laundry list of antibiotic resistance genes. Now, let's say for the sake of argument, you zoned out for the last 2 slides and didn't really care about the technical details here. The main take-home point is you can take a method of this type and apply it to any arbitrary group of microbes and use it as an engine to discover cryptic antibiotic resistance genes, as well as known ones that are compatible with a pathogen and will model horizontal gene transfer, and then will tell you what the identity of those genes are. It's a discovery engine. So how do you apply this discovery engine to understand something more about antibiotic resistance biology and ecology? So first off, I'll take a quick break here to just tell you that resistance spreads across habitats. I can show you all sorts of fancy schematics, but this questionable parenting event perhaps captures that idea that microbes can talk across habitat barriers better than most slides. But let's go back to the perhaps more scientific version of this to then zoom in for the rest of this talk on a habitat that is sometimes not considered as prime in the transmission network, but I would argue is very important to consider in the resistome, and that is the resistome associated with soil microbes. So why look at the soil microbiome? We could give a whole lecture series on this. I'm only going to highlight 2 or 3 pieces of work by others that brought us to this particular set of questions. The first was this work by Jerry Wright's group about 10 years ago with this very informative title, Antibiotic Resistance is Ancient. It captures this idea that they went into the Beringian permafrost in Canada, carbon dated it to be 30,000 years old, and then when they sequenced DNA from these samples, they found resistance genes against modern antibiotics, providing clear genetic evidence that resistance clearly predates any kind of modern human use of antibiotics. The other is this producer hypothesis put forward by Julian Davies and colleagues now almost 45 years ago when it was originally talked about. And that recognizes that these so-called magic bullets that we use as antimicrobials in the clinic are largely natural products of the soil streptomyces. And the producer hypothesis argues that since these production capacities in the soil are millions, if not billions of years old, they've given enough selection pressure for those producer organisms and all of their neighbors to have antibiotic resistance. And why the production organisms? Because antibiotics target the most key features in bacterial life. And these bugs would basically commit suicide if they didn't have just-in-time resistance. And then when you look at data that is available, and in this case, the Graham group looked at soils over the course of the human use of antibiotics, and these are soils in Europe, they noticed that important antibiotic resistance gene categories had levels that increased over that correlated time. And so that is arguing that, and this is a correlation, not causation, but at least suggesting that if you take these other pieces of information into account, that we as humans and our use of antibiotics have caused a concomitant increase in antibiotic resistance genes in the environment. So with all of this put together, the one hypothesis that you might make then is that antibiotic resistance genes that we have in the clinic, in clinical pathogens, come directly from these soil organisms. These are the original evolutionary progenitors of all antibiotic resistance. And that's well and good, but how do we actually prove that? How do we provide genetic support for this idea? Are they indeed in recent exchange with pathogens, even if they had actually donated this millions of years ago? And so to answer this question, we first looked to the literature about 10, 15 years ago, and we were quite surprised that we couldn't find too many resistance genes described in pathogens that were genetically identical to those that have been described from soil organisms. And the reason I say genetically identical is because that's kind of our clock for how recently an exchange occurred. If I find the same gene in two organisms, I can say it's quite likely that they exchanged that recently, or they at least had a very recent common donor. We didn't find that evidence, so we went hunting for that evidence with the hypothesis that perhaps it was a needle in a haystack problem, and we just needed to identify the minority members, if you will, of the soil microbiome that might be that missing link. So to do that, we performed a sort of strange experiment and actually harnessed a strange experiment, which I'd actually started when I was a postdoctoral fellow, where we happened to have selected for microbes from 11 different US soils in this case, that could eat antibiotics at exceptionally high concentrations. So these 1,000 micrograms a mil is about maybe 50 times the concentration that would define the killing concentration of most of these antibiotics. And weirdly, these bugs not only could resist them, they could utilize them as sole carbon sources. That's a different story, but we decided that this perhaps was a way in which to refine that bottleneck. Look for the most hardcore resistant bugs in the soil, and perhaps they would have the kind of resistance genes that cause bugs in the clinic to be resistant. And so what we did is we took this collection of bugs from 11 soils in 18 different antibiotics with replication, and enrich them such that we could then look at their metagenomic DNA, and then apply this particular selection scheme called functional metagenomics. This is work led by Kevin Forsberg and Alejandro Reyes, prior graduate students here at WashU. And what they did was they found this particular evidence. So by applying that engine to this unique bottleneck of bugs that were highly resistant to drugs, they were able to find resistance genes that were 100% identical from those soil organisms to a variety of different pathogens that have been found all around the world. So even though this was a pretty small assay in retrospect, it was surprising that we found 10 such resistance genes that represented resistance against 5 different classes of antibiotics, all of the major known mechanisms of resistance. They were identical to genes and pathogens that were some gram positive and some gram negative. And shown in this map of the world are in dark the countries where those pathogens had been deposited, where the resistance genes were identical. So even though this was an assay we ran on US soils, we were discovering something that was perhaps global. Okay, and this shows you a comparison to really suggest the problem is even bigger than we originally anticipated. What this shows you are genes that come from our selections from a variety of different antibiotics, and these are from our soil selections, and compared against, at that time, a database of known pathogens. And we found was not only were we finding these genes were identical to genes and pathogens, but they were clustered together. So we were discovering that actually it was not a single resistance transfer that perhaps had occurred in either direction, but rather clusters of these resistance genes that had somehow gone back and forth. And the reason we believe that these are true clusters is we can not only look at the resistance genes, which are shown here in red, but you can also look at these interesting genes colored in yellow And these are mobilization elements. These are one of the engines through which bacteria move DNA back and forth between their genomes. So we're finding, at least again, genetic signatures here that there is clusters of antibiotic resistance genes flanked by the things that move them back and forth that are identical between soil organisms and pathogens. So now providing clear genetic evidence that this does occur, and it likely occurs over very short timescales. Okay, so that's the observation, right? So we run this kind of bizarre assay in terms of enriching for bugs that can eat antibiotics from the soil. We find that they're multi-drug resistant. We actually identify that most of them belong to this proteobacterial phylum, and that we're providing genetic evidence that they had recent exchange with pathogens in the clinic. But is it the exception or the rule? Is this weird selection that we did representative of the entire soil microbiome? And the reason you care about this is it's for risk analysis. And so in this case, Kevin, who you saw his name before, teamed up with Sanket Patel in the lab to ask this question. And so to do this, we collaborated with two labs, Noah Fearer's lab and Rob Knight's lab, at that time both at the University of Colorado, to access soil samples that have been carefully curated and collected from two different states in the US through these long-term ecological research networks from Minnesota and Michigan. And I won't belabor the details about everything about those particular soils, except to say that Rob's lab and Noah's lab had already collected information about what bugs were in those soils. So that we so-called 16S phylogenetic sequencing. So where they sequenced a particular marker gene in those samples and said, here's the catalog of who's there. And so what we did was we went to those same soil samples and asked, what are the resistance genes in there using that functional metagenomic technique? And what we found was, interestingly, and don't have time to go through all of the math behind this, but this is a method called a Procrustes analysis that allows you to compare two different data types from the same sample. And the closer two dots are here, the more related they are. And so one dot represents, in this particular two-dimensional representation of an n-dimensional space, the taxonomic data, who's there, and the other dot represents their resistance genes. And what we found was very, very tight clustering. So that is, for all of those particular soils, we found that the bugs who were there are somehow connected to the genes that they have with them. So why do I bring this up? So from this, we can make one particular type of inference, and that is to say, perhaps in this case, even though there are a lot of resistant genes, we found 3,000 unique resistant genes in this particular assay, maybe most of them are not actually amenable to horizontal gene transfer, unlike what we showed in the prior assay, right? The reason being is that if I'm a particular bug and I have another bug that's a particular distance away from me in terms of our genomes, if I donate my gene over to that other bug, that immediately that gene is going to look more like me than that other bug. So if you were to sequence that other bug now, you wouldn't see that tight connection between that recently acquired gene and its genome. So that's one way to infer, if you find genes that don't quite look like the other genes in the genome, that perhaps is an example of horizontal gene transfer. That doesn't seem to be the case with most of these genes. And so to verify this, we did something similar to what we had shown you before. We started looking for resistance genes and the genes around them, to argue that if there were genes that were in these soils that were clearly giving resistance, but they were not easily amenable to transfer, then you wouldn't find them as often associated with those mobilization elements that I told you about before. And we just counted those frequencies. So we said, let's go to a bunch of soil genomes, let's go to a bunch of pathogen genomes, let's identify the resistance genes using a computational method, and look how frequently they're next to mobilization elements. And sure enough, this particular hypothesis was supported by that analysis. That is to say, when we look at genes and pathogens that are resistance genes, they're way more frequently associated with mobilization elements than most of these genes that we find in soils. So how do we reconcile these two stories then, right? What does it mean? It's almost contradictory. So from this, we have the following model. We find, consistent with the producer hypothesis, that the resistome in the soil is huge. In fact, probably per capita, there are more resistance genes in most soil organisms than your average human-associated bug. But because the selection pressures are likely very, very old, they're not necessarily hot swapping them at any high frequency, at least in comparison to pathogens. But there is a tiny, exceptionally important minority. In this case, it appears to be the proteobacteria that appear to have the ability to do those kind of transfers. And that's also consistent with evidence from hospitals, for instance, right? The proteobacteria happen to include a lot of organisms that are cosmopolitan in terms of the ability live across lots of different habitats. They can live in the soil, they can live in humans, and they can also live in a hospital, ventilation systems. So because of that ability and because of the ability to go through this horizontal gene transfer, they might be the ones that through this huge milieu of bugs and genes in the soil that we may want to specifically focus on. Okay, so then I'm going to end with saying, okay, that's an ecological view, but how do you as a committee use this information to say, how could you use methods of this type to actually do something actionable in the clinic? So this is a story about tetracycline resistance that comes directly out of the study I just told you about. So why tetracycline and why tetracycline resistance? Tetracycline happens to be one of the largest class of antibiotics used both in the clinic and in agriculture. It was discovered in the late 40s. It was part of the heyday of antimicrobial discovery from the soil. And because of its huge deployment, it makes sense that resistance arose and continues to go through for sort of hand in hand. And the two major modes of resistance against tetracyclines, and pathogens at least, appears to be either to pump the drug out through active efflux, or to have proteins, these cleverly named ribosomal protection proteins, because they protect the ribosome from the attack of the antibiotic. Now, interestingly about these two mechanisms is neither of those mechanisms do anything to the antibiotic itself. You get the drug out of the cell, here you protect the target of the cell, but the molecule of antibiotic is still around. And that seems a little bit interesting because you're then missing a very critical option in terms of destroying the antibiotic as the bug, which will help you and your neighbors. And this mechanism is called enzymatic inactivation. And up until around the time that we started looking at this in the sort of mid 2010s, there was only one unique gene that had been shown to have this activity, and it was not in a pathogen. It was called TETX. It had been found in a human gut bacterium. And there were a couple of variants that had also been described, but it had this unique property of being able to destroy the tetracycline structure itself by first hydrolyzing it. So Kevin shows up again, and now this is a collaboration with a chemist here at WashU, Tim Vincevich, where through his various functional selections, Kevin looked for the opposite of what we usually look for. He looked for genes that were coming up through our functional selections that were not recognizable antibiotic resistance genes, yet were providing antibiotic resistance activity and were coming up over and over again. So it's a simple sort of human pattern recognition exercise. So you notice that in a lot of our tetracycline selections, this kind of vague annotation of FAD-dependent oxidoreductase kept coming up, and there were no other recognizable tetracycline resistance genes around that. And so he did a lot of work to clone these genes out. He speculated that maybe they worked like TETX, even though they were not sequence related to TETX. Sure enough, when they cloned them out, they gave resistance to E. coli at high levels. This is showing you the alignment of our 10 sequences against TETX and a couple of its neighbors. And again, don't have time to go into the details, but just based on distance on your slide, you can notice that these are much more closely clustered together than these three. So they're not recognizable trivially by sequence. The only gene in the databases at that time that we could confidently say looked like any of our genes was a gene that was found in Legionella. Legionella, a soil-derived pathogen, causes Legionnaire's disease. And fortunately, we happen to have a Legionella pathogenesis expert at WashU, Joe Vogel. And through some work with Joe, we were able to show that even this cryptic resistance gene, this gene that had no known resistance identity in Legionella, by identifying it using this method, we actually identified that it is a tetracycline resistance gene in Legionella, which coincidentally is one of the drugs that Legionella disease had been failing against. And then we did a series of assays, which I won't go into, this is all published, to show that, in fact, that is the mechanism of resistance, that these are enzymes. They are breaking down the antibiotic using colorimetric assays and mass spectrometry. We could show that they're decreasing the concentration of the drug, not just simply getting it out of the cell. Okay, so then another grad student, an ex-grad student, Drew Gasparini, said, okay, well, we stumbled across this, or Kevin stumbled across this across those 10 soils. That's where all of that data came from, that same study that we had done from soils from Noah and Rob's labs. And Drew said, well, our lab does these functional selections all the time from a variety of habitats. Let's go hunting for these particular genes in everything we've done so far, right? And that is to say, we've done tetracycline selections from soils and human guts and latrines and all sorts of other things. Let's go and see, do we find those FAD-dependent oxidoreductase, that weird annotation? And sure enough, just from that scan, retrospectively of our own data, Drew was able to predict that we probably had another 70 unique such enzymes in our freezers. And again, just to put this in perspective, right? There was one such enzyme that had been characterized before this. Kevin found that there were 10 more in the soil. And Drew just retrospectively goes and finds 70 more, suggesting that there is something going on here that for some reason we've had our blinders on and not noticed. And unfortunately for humanity, but sort of, I guess at some silver lining, fortunately for us in terms of having this bellwether event, exactly when this was happening was when TEDx, that original human-derived enzyme, started popping up into important pathogens. So we had certainly speculated that this could occur, but we were surprised that it happened to occur right around the time that so we somehow happened to catch this bellwether event, right? So that was our now motivation. We've got this sort of lurking under the surface enzyme class that seems to be all over the place. It was undetected until now, but now it is popping up all over the place. So we decided, let's take a basic science approach. Let's throw the basic science kitchen sink at this problem to understand this enzyme mechanism, to get better at predicting it, and ideally, by being a little bit ahead of the curve, by being proactive as opposed to reactive towards antibiotic resistance, perhaps we could even defeat it. So one of the things that Drew did then was he took these enzymes, which are from the soil in the top, and he compared the resistance that the new human enzymes, 10 of them that he found, encoded against the tetracyclines. And what he was really surprised to find is that much like the separation that we found in sequence, that is these soil-derived enzymes by sequence look quite different from the human gut-derived enzymes. They also end up correlating with differences in phenotype. So what you're seeing here is from light to dark is the darker the color, the greater the level of resistance that this particular gene is giving to E. coli when expressed in it. So it appears that all of these soil enzymes tend to knock out these first couple generations of tetracycline drugs. So these are organized by generation of when they came to the clinic. But the gut guys also knock out now the third generation drugs. And the reason this is really important, I've highlighted here 2 drugs that we actually got a hold of before they got on the market. So this was approved in 2018, but this work, and certainly the samples were collected at least 4 or 5 years before that, to really emphasize this idea that antibiotic resistance enzymes are lurking in wait. And as soon as you apply selection pressure, they're ready to activate, right? So that's the hypothesis here. So that is why, even though these enzymes have probably never seen omadacycline and arabicycline because they've not been approved for by the FDA yet, they yet had perfect ability to blow them up just as well as they blow up the earlier generation tetracyclines. And that's why we think actually this selection pressure might be driving. That is, the introduction of newer drugs are now going to activate the necessity for these enzymes to come in and perhaps take over as a dominant mechanism of resistance. So that's all of the what's bad. So what can we do with this sort of bad news? And so in this case, we brought on another collaborator, Neeraj Tolia, who used to be in microbiology, now's at the NIH, and worked with his graduate student to try to say, could we defeat these enzymes? And so we went back to something that's pretty well established in the clinic through basic science. And that is to say, one way in which you can defeat enzymes that break down antibiotics is to find decoy molecules that can go and gunk up those enzymes, so-called resistance enzyme inhibitor. So this is very well known with the beta-lactam class of drugs, where you can find compounds that look like they look similar. They have this similar kind of beta-lactam warhead, but because they don't have this other decoration, they themselves are not antibiotics, but they look like them, and so they trick the enzyme and they become suicide inhibitors. So we decided, let's go looking for this using the same feature. Let's look for compounds that look like tetracyclines, but are not necessarily antibiotics. And this is the part where I usually caution graduate students in the audience that don't take too much away from this, because we got dumb lucky that the very, very first compound we tried happened to work, right? I don't think this is exactly how it normally works. We were lucky and we will go with that luck. And so what happened was Drew happened to pick anhydrous tetracycline as a potential inhibitor and found that it does exactly what we wanted it to do. So what you're seeing here is a UV trace that is looking at an enzyme reaction, an in vitro enzyme reaction. We're incubating the enzyme with tetracycline, and the tetracycline peak time going up disappears very quickly because it's being chewed through. But when we add anhydrous tetracycline into that reaction mix, tetracycline peaks are protected. And this was true against all of the enzymes, at least in vitro. So that's exciting news. We now happen to have a candidate molecule that can gunk up the activity of this enzyme against tetracycline, okay? Now, because we want to really go at this to understand how this works, Neeraj's lab, and JP in particular, solved a series of beautiful crystal structures to begin to understand at the molecular level, how does this work, right? So that's the pitch here. Why do you want to do this kind of thing when you've already described the enzyme? Is to say, if we understand how this works, we can break it. right? And so I'm not going to go into all of the details. These structures, they illuminated a series of interesting things about the enzyme mechanism. But the important part, and again here the fortunate part, was JP was able to solve a structure of some of these enzymes, both with the substrate, so the tetracyclines, as well as the inhibitor, to give us a molecular understanding of how the inhibition might work. And so what did he find? So he found a couple of different things. And again, I don't want to go through every one of the details, except to say through this structural biology work, we're able to come up with rules now as to why the inhibition might be working, such that we can design new inhibitors. We found things about how they were in different conformations, but again, we can go about this offline. But the important part is, by applying molecular methods, we're able to understand why, perhaps serendipitously, something we find, why does it work? And so we've been using this now with this trifecta collaboration between the Vencevich, Tolia, and my lab to understand these enzymes better, to go and try to understand how they work, and to design inhibitors. And so interestingly, as this was going on, we and others are continuing to look for new enzymes in terms of where they came from. And I already told you, in terms of this connection between the soil and the environment, we had already in our very first work identified a connection between one of these soil bugs and Legionella. Well, as it turns out, as Drew was looking through human enzymes, we also happen to, in other work, be working on infectious bacteria from a variety of different places. And from a cystic fibrosis patient from Pakistan, we happened to identify a Pseudomonas aeruginosa isolate that just so happened to have exactly the same enzyme. So again, a bit inadvertently, through our searching, we already found one of these missing links. So a soil bug, which is identical to another soil bug, which happens to be a pathogen, a human gut commensal, which happens to have the same enzyme as something that's in a pathogen. So again, this is not a question of if resistance is going to occur and get out. It's already happened. And we just happen to be discovering it, hopefully a little bit early. And we were not the only people looking at this. As part of the One China initiative, there are a variety of, in a sort of parallel investigation, a variety of reports over the last couple of years that these TEDx type genes were popping up all over the place. And these couple groups did some really nice work to show that these enzymes were now found in conjugative plasmids. So now they were in the context of the type of elements that move them back and forth. They were in some of the worst types of nosocomial infectious bacteria that are these cosmopolitan organisms. And these groups isolated them from humans, from meat for consumption, and in animals. And they also did animal work to show that in a mouse model, these resistance genes would compromise tigercycline therapy against an infection. So really showing all the way through, they're there, and they can be a really serious problem. Okay, but so here's a little bit of good news then. As I mentioned, we've got these latest generation tetracyclines coming up. They happen to be one of these classes of drugs that actually are seeing a bit of a renaissance in terms of reactivation. And so through work with the Vencevich lab in particular, they made a series of chemical modifications through synthesis of a variety of different anhydrotetracycline backgrounds. Remember, that's our original scaffold. And what they were able to show now is that through a lot of hard work on the organic chemistry side, they were able to come up with variants of anhydrotetracycline that have activity not only against the enzymes themselves in vitro, but also when we put them against bugs like E. coli or our original Pseudomonas aeruginosa isolates. And they allow those bugs now to be killed again by the original antibiotics, right? So again, this is the story of how you can go from a bit of a serendipitous discovery like anhydrotetracycline by working with experts in organic chemistry and biochemistry, able to modify those compounds so that they can reactivate these particular drugs. And then I'll just end here to say, this is kind of where things stand, right? Because of this discovery we made that was really very ecologically driven, right? We were initially looking to find patterns in that early paper by Kevin of how resistance genes are distributed across environments, and stumbled across an enzyme class, which we call the tetracycline destructases, right around sort of this point. Right? And what you're seeing in this top plot are all of the unique resistance genes that are described in terms of the three major classes of resistance I told you about before: efflux, ribosomal protection, and now this tetracycline destructase, right? So here you've got many decades of time where these particular efflux pumps and ribosomal protection proteins, in terms of the number of unique resistance genes, keep coming up and spreading. And then lo and behold, here is when we happen to discover these soil enzymes. This is when that original TEDx had been discovered. And now the number of those unique enzymes, in terms of our knowledge, is beginning to approach the same number as the ones that we had seen in these other classes. And we think the easiest explanation for this is a combination of us looking for it using the right approaches, but also we as the world are beginning to provide the perfect selection pressure through tetracycline, omadacycline, arabicycline, and saracycline, these next generation drugs, which effectively require the action of these enzymes to be defeated, because those new classes of drugs, these latest generation tetracyclines, are mostly immune to the other mechanisms of resistance. So as soon as you bring in that selection pressure, you're going to see this flip is what we're predicting in terms of those resistant enzymes. So I'm going to end there, right? Just to say, hopefully what I've shown you over this presentation is by taking this ecological view of antibiotic resistance, by looking at the resistome, not only do you begin to understand patterns of where resistance came from originally, right, in terms of the connections with the soil, but it also gives you these opportunities for detecting bellwether events, for finding cryptic resistance genes in interesting ecosystems, but then you still need basic science approaches to go through, understand how they work, and if you happen to get lucky, also identify ways in which to defeat them in sort of real time, right? An ability to, so that we now have inhibitors that could be used in tandem with these particular drugs to rescue their activity before they're defeated against these latest class of enzymes as they're beginning to spread. So with that, again, I'll just remind you about this idea that, we should be careful about thinking that our habitat boundaries are protected. Microbes don't care. Selection pressure in one environment will certainly transmit over to others. We don't have time to go through this, but we have ongoing and published work in every one of these particular habitats. And what we're really hoping to do is take the type of methods I just told you about, as well as other computational and experimental methods looking at the bugs themselves, and beginning to put, rate constants, if you will, on these transfer events. So these are cartoons right now, but we would like to say, if I was to look at this arrow from soil to river versus this arrow from river to people, which one is at greater risk? And we think we have methods now by doing the discovery engine along with the prediction engine, especially with longitudinal analysis, we can begin to do this. And where we've been perhaps most successful in doing that in our lab is actually in the human microbiome, looking at this interface between especially the bugs in the gut of humans, which are constantly under antibiotic selection pressure, and the genes that traverse, especially bugs that cause gut-derived infections. So I end there, just tell you that, this is our pre-pandemic days when we were able to not socially distance. This is really what our lab looks like right now, like this particular call. I'll end with certainly thanking this incredible group of people who I'm really here as their representative, both currently as well as the people who've gone on to bigger and better things. None of this would be possible without incredibly fantastic collaborators, both domestically and internationally. And then, of course, we're very, very grateful to the various federal as well as private funding agencies that have been kind enough to help us do some of this work. So I'm happy to take, questions from the committee if that's appropriate, and end you with just this particular PSA, given that we're in the midst of this viral pandemic, that please do not use antibiotics to treat viral infections. And hopefully that's rhetorical for this crowd, but it's my opportunity to proselytize. So I'll end there.
Superb. We have just a couple minutes for questions. Lefteris has his hand up, so let's go with that one. If we have time for another one, we will take it.
Hi, I have two questions. First one, great presentation, by the way.
Thank you.
Enjoyed it. How is global warming going to affect environmental resistome? Do you see any trends?
Yeah, sorry, go ahead.
And also, with such a vast resistome, is there any chance that we can hope for molecular diagnostics to help with real-time treatment of patients? Thank you.
Yeah, excellent questions. We don't have direct work going on the global warming question. So I'll just say my intuition is that, as we perturb the environment has reasons to respond, if you will. And I think, where we will see differences are the activation of microbes that are in habitats that were previously inaccessible that might come into the milieu. But I think the greater impact of global warming is we're going to have more and more vulnerable communities that are going to be put into situations where they're at greater risk for infectious diseases. So I think for me, that's the greater risk. In terms of your second question, in terms of what do we do when it's so overwhelming out there, right? How do we get these needles from a haystack? I think we shouldn't be overwhelmed by this vast resistome because we've existed with this vast resistome for a very long time. I think we know that most microbes are out there likely trying to protect themselves, right? These are just events in terms of their toxin production mechanisms. And what we need to focus on are those microbes that have the ability to break out of their barriers, to get into our hospital systems, and to track those and to come up with methods to understand, for instance, can we, as we're doing surveillance, look for those genes that appear to be in the type of bugs in the environment that could get into us and also appear to be around these mobilization elements. I will, however, just emphasize that we're always going to be playing catch up to some degree. There is not going to ever be a 100% predictive regime because evolution is at play, right? So I think we can do so much, right, in terms of antibiotic resistance prediction, but we're always going to have our discovery engines on to be able to keep a little bit ahead of the game.
I think we have time for one more. Erica, you have your hand up.
Yeah, thank you. Thank you for the talk. It's a really nice talk. My question is actually very specific. It's about anhydrous tetracycline. And I'm just wondering why. So anhydrous tetracycline is very common in nature. It's one of the most common breakdown products of tetracycline. And even today that we use tetracycline clinically, you can find it in tetracycline capsules that are being sold in the sun, which is pretty common in my environment. So I'm just wondering, why was there this lag in terms of selection? I mean, why didn't the selection for the, why didn't the selection happen before omadacycline and so on came onto the market? If there was a natural molecule that was abundant in nature as well as in clinical settings as breakdown product in poor quality tetracycline preps.
Yeah,
why do you think that it didn't offer the selection that these new tetracyclines are offering now?
Yeah, that's a wonderful question and one that we're actively trying to pursue. We don't have a direct answer to that, right? So here's our speculation though. So we think that, like all of these other resistance mechanisms, there's a lot of potential out there in terms of environmental resistomes. And this is just one example. And part of it could simply be that it's a bottleneck. It's an evolutionary stochastic bottleneck, that there were multiple mechanisms at play. The first mechanisms that happened to get into the type of bugs that cause infections were efflux and ribosomal protection. And there was a founding effect, and that just took over. And while the enzymes were available, perhaps they were not necessary, right? Because head to head, it appears that the enzymes don't necessarily provide any additional benefit in terms of resistance against the earlier generation drugs. So under that hypothesis, it was just a mistake, a happy mistake. And then it turns out that now that you're providing the selection pressure, the mistake, the tables begin to turn a little bit, right? So that's one hypothesis. Another intriguing hypothesis, one that we're trying to experimentally test, is that perhaps there's actually a fitness, there's some kind of fitness challenge that the enzymes have, that they're actually not beneficial to have around, or at least on, right? They give you some kind of negative impact in terms of carrying it around. And so we think that there are reasons to believe that that might be true. So if you do a head-to-head competition, as an example, between an enzyme expressing E. coli and an efflux expressing E. coli and a RPP expressing E. coli, which are exactly the experiments that are going on, that's what we're going to test. Maybe the enzyme guys, if they're always on, they get killed off. And we think that there are some mechanistic reasons for that. There's also some really interesting regulation that we don't have time to get into, whereas it turns out, this is what's beautiful and bizarre about the bacterial world. Those inhibitors, anhydrotetracycline that we show that can inhibit the enzymes, also appear to be the same molecules that activate the pumps, right? So they have the ability to go on and turn on efflux pumps. So there's a lot of weird stuff going on. We're at the beginning of trying to figure it out, but that's partly what keeps us up at night, and it's a wonderful question, something we'd love to understand.
That's really cool. Your answers are really interesting. Thank you.
Of course.
Yeah, thank you. Really appreciate it. I think we could probably have a dozen more questions because it's such an interesting area. We don't have time for that, unfortunately. But I want to thank you for your time this morning and really appreciate it.
Of course. Thank you. Thank you for what you guys are doing. Thank you, Jillian, for the invitation. Michael.
And to the other members,
good to see you. Always a pleasure.
We're gonna actually close and then rejoin at the top of the hour for the closed section.
See y'all.
Thanks,
Okay.
folks.
Super.